I'm Srijesa, and my project is called Exploring Intrinsic Hippocampal Contributions to Spatial Working Memory, which I know sounds like a mouthful, but we'll go through it slowly and it'll all make sense. So the hippocampus is a brain region that's important for memory consolidation with respect to time and space. And it's a bilateral structure, meaning it's in both hemispheres of the brain. So here on the right, we're looking at a slice of a rodent brain, and you can see the hippocampus is in both hemispheres. And then if we zoom in, we can see that there are different divisions of the hippocampus, such as the dentate gyrus, CA1, CA2, and CA3. And the hippocampus is important specifically for episodic memory, or the memory for specific events and when they occur in time. And this includes things such as what did you have for breakfast and your morning routine. And it's important for forming larger sequences of events. One of the most famous case studies in this field is that of HM, who received a bilateral resection of medial temporal lobe structures because of uncontrollable seizures. So this just means they took out parts of his brain, and one of those parts included the hippocampus. And since then, researchers had been studying him and administering different clinical tests to see what kind of deficits he was experiencing in memory. And those included anterograde amnesia, deficits in long-term declarative memory, as well as the inability to create new episodic memories. So this could have pretty devastating consequences in your day-to-day life. So the kinds of cells that I'm looking at are called time cells. In general, in the Eichenbaum lab, we study memory by training rats in different tasks and then recording their electrical activity using a device called a Hyperdrive, which I'll get into. But using this method, we're able to identify different cell types and put together some pieces about how memory works. So one of the common cell types is place cells, which are known to fire in specific locations within an environment. So if you look at this task here on the right, it's just the rat runs around the track, and we see different cells fire at different places. So these purple ones are firing more at the center, these orange ones more at the bottom right. But in addition, more recently, we started working with time cells, which are known to fire during a delay in a memory task. And these have been identified in the CA1 and CA3 regions of the hippocampus. So we expect to find time cells on a paradigm like this called the delayed alternation T-maze task. So we start by putting the rat on the treadmill for a number of seconds, and the rat receives a water reward before and after the treadmill run. And then he's in the center stem, and as long as he takes alternating right and left turns, he receives water rewards from the respective ports. So on the right here, we're looking at single cell activity during the treadmill run, and this is where we'd expect to see time cells rather than place cells because there's no change in external cues. The rat is in the same position, so the only thing that could possibly be changing is how the rat is keeping track of time. So on the right, we're looking at time on the x-axis, in this case 15 seconds, and then cell firing on the y-axis. So up here we have a rasterogram where each dot represents the cell firing, and that's summarized below by this frequency curve. So you can see that this cell in particular is firing more near the beginning of the treadmill run, whereas this one prefers to fire kind of in the middle, and this one is near the end. So these time cells are firing preferentially at certain time windows during this entire delay in the task. And this is all kind of compiled in this normalized peak firing rate graph on the right, where we're looking at time again on the x-axis, but different cells on the y-axis. And red denotes the highest firing rate, while blue denotes the lowest. But overall, we're seeing a clear sequence of cells firing. And this is just a simplified circuit showing how temporal information flows through the hippocampus. So information goes from the entorhinal cortex, which is a region outside the hippocampus, and it's relayed to different regions of the hippocampus, so dentate gyrus, CA3, CA1. And for the purposes of this project, we operationally defined this pathway from entorhinal cortex to CA1 as the extrinsic pathway, because information outside is going in, whereas CA3 to CA1 is defined as the intrinsic pathway. And as I mentioned before, we've seen time cells in CA3 and CA1, and recently an experiment in the lab showed that getting rid of these extrinsic inputs caused a loss of time cells in CA1. So the next logical question that we wanted to ask is, what would happen if we got rid of these intrinsic inputs? So that brings us to the project description, which will make a little more sense now that you have some background. So we're trying to understand how a transient disruption of intrinsic hippocampal circuitry could affect temporal processing, and this will help us understand the dynamics between extrinsic and intrinsic pathways and also how they influence time cell activity. So in terms of our methods, as I said, we used a device called a Hyperdrive to record neural activity. So in this device, there are 24 individually drivable tetrodes, and a tetrode is just a very fine wire that we can lower precisely into the brain. So there are 24 of these little things called tops, and each turn corresponds to the tetrode being lowered a 1/4 millimeter into the brain. So using this, we can pick up the changes in electrical activities in the cell. So this is the device, and this part is implanted into the brain, and this upper part, called the EIB board, is connected to a computer, which allows us to visualize the data in real time. So the kinds of data we see are just these different waveforms, or the cells firing. And this is just a picture of an implanted rat, if you want to put together the pieces of how this all works. But the method we're using is called VHC stimulation. So the VHC is the ventral hippocampal commissure, and it connects both hemispheres of the hippocampus, but it also connects CA3 to CA1. And basically, we have a fixed wire in these fibers that connect CA3 and CA1, so we can provide the stimulation, and it's very transient on the time scale of 0.1 milliseconds at a very low current that only disrupts the hippocampus. But we're also simultaneously recording from CA1 with the Hyperdrive. So this data is just showing that we were able to successfully use this method and not damage the brain. So in figure B, we're looking at the activity of a population of cells, and you can see that the cells are firing consistently. We're looking at voltage on the y-axis here, and with our disruption, you see a change, but also recovery. And same goes for figure C, where we're looking at the theta phase of the local field population, and this is just a rhythm that time cells are known to fire with. And we see again the silence, but also recovery, and the last figure is just single cell activity. So again, this is the task, and the stimulation paradigms are as follows. So we have our time stimulation. So this just means within one session where we work with a rat for 50 minutes and it runs 50 trials. The first 20 would be our control, the next 20 would be the time stimulation. So we provide the stimulation while it's running on the treadmill, and then followed by 10 for recovery. And then the play stimulation is just our control, because past experiments that have used this method have shown that play cell activity remains intact. So in terms of our results, so far we collected data from 5 rats, but we've only analyzed that from 2. And of those 390 cells we saw, only 67 met the criteria for a time cell. So here we're again looking at time on the x-axis, which was 8 seconds in our experiment, and then cell on the y-axis, and yellow denotes the highest firing, blue denotes the lowest, and you can see that there was this clear trend of cells firing. But then we looked at how this same set of cells in this order were behaving in our different conditions, so in stimulation and recovery conditions. And we saw a loss in this clear sequential activity. So using this manipulation, we're disrupting something about these time cells. And we further quantified that in this histogram, where we're looking at firing rate per second on the X and cell on the Y. So we're comparing the activity of these same cells during the baseline conditions and post-baseline conditions. So if we were to see most of the cells around 0, that would mean there was no difference. But since most of them are kind of in the left, we can say that there was overall decreased peak firing rate of these cells. And we did the same thing where we defined the cells in the stimulation condition. But when we look at these same cells in baseline, we don't see any kind of activity, and recovery is not the best either. But lastly, we defined time cells in the recovery conditions, and we saw this return of like a clear sequential pattern of these cells firing. These same cells weren't doing anything significant at baseline conditions, but in stim conditions, we do see somewhat of a trend. So what this implies, because between the stim and recovery conditions, that's when we restored, or these inputs to CA1 from CA3 are restored because the effect of the stimulation had faded. So restoring these inputs is somehow contributing to time cell stability in CA1. So what does this mean? Well, first of all, time cell activity persisted, but we saw decreased cell stability, so changes from baseline to stim, but not so much from stim to recovery. And the preliminary conclusions we can make is that with transient CA3 to CA1 disruption using VHC stimulation, CA1 time cell activity is modified from baseline, but it persists. And we also did other sets of analyses looking at things like information and sparsity, so just different characteristics of time cells to quantify how they were changing. And we noticed diminished peak firing rate, a loss of temporal information, and an increase in sparsity. And so for future directions, we'd want to look into things like theta and what happens if we change the time of stimulation, as well as other paradigms, so maybe providing randomized stimulation rather than consistently providing it across 20 trials in a session. So I just want to say thank you to everyone at the Eichenbaum Lab who has helped me with this project throughout the years. And any questions? Yeah.
Do you use male or female rats for your experiments, and is there a reason for picking specific cells?
So in the lab, we always use male rats, and I think that's because they're somewhat easier to work with, at least from a behavioral standpoint. So the task can get pretty complicated because we're first training a rat on a treadmill, and then we're asking them to do, so they're water deprived, so they're motivated to do the activity, but the treadmill training is probably the hardest part of the task and getting the rat to stay on that consistently. So I think just for behavioral reasons, most of these experiments use male rats, but I haven't really read anything on the female rat behavior. All right. All right. Yeah. Thank you, everyone, for coming.